Thanks for the introduction. Good afternoon, everyone. I'm Zhaoyue Shi from Vanderbilt University. As we know, FMRI studies rely on detecting hemodynamic changes to infer underlying neuroactivity, which is an indirect indicator of actual neuroactivity. Resting state correlations between both signals are interpreted as functional connectivity. On the other hand, local field potentials refer to electric potentials recorded in the extracellular space around neurons. They relate well to processes in dendrites and mostly represent the inputs to a particular brain region. Therefore, local field potentials are considered more direct indicators of neuroactivity than both. We need to know more about the spatial and temporal relationships between changes in neuroactivity and changes in hemodynamic. The validation of neural basis of FMRI signals is essential for interpretation and application of FMRI-based measurements. Therefore, we aim to quantitatively compare the time-varying patterns of resting state function connectivity between BOLD and LFP signals in primary somatosensory cortex using a sliding window technique and Markov model-based approach. We acquired both tactile stimulus and resting state BOLD FMRI signals from squirrel monkeys at 9.4 Tesla. This red box shows S1 cortex, which can be identified by some landmarks, for example, central sulcus and lateral sulcus. S1 cortex includes areas 3A, 3B, and 1. Here, we mainly look at areas 3B and 1. We used the stimulation to identify ROI seeds for resting state analysis. In our stimulation protocol, each stimulation consists of a pre-stimulus period of 30 seconds and a stimulus presentation period of 30 seconds. In both FMRI, our in-plane resolution is 274 microns. In LFP experiments, we inserted two multi-channel electrode arrays in areas 3B and 1. There are 98 electrodes in total, and the spacing is 400 microns. LFP signals were sampled at 500 Hz, and then bandpass filtered into 8 different frequency ranges, such as broadband, delta, theta, alpha, beta, gamma low, gamma high, and gamma very high. I would like to give a brief summary about our recent published results on the spatial relationship between BOLD and LFP. The broadband LFP. We found the average distance of the activation locations between BOLD and LFP was around 0.35 millimeters, which means the activation locations between BOLD and LFP agree with each other very nicely. We also found comparable spatial profiles of resting state BOLD and LFP functional connectivity in areas 3B and 1. Resting state correlation profiles are slightly wider than activations. Our following work is based on this one. This work is done only on the broadband LFPs, and here are the results for spatial extents of 7 frequency bands. We compared the area values between resting state and stimulation and found that in area 3B, in low frequency LFP signals, the spatial extents of resting state are significantly wider than those of stimulation. However, in high frequency LFP signals, the spatial extents of resting state are significantly smaller than stimulation. In area 1, we can see the similar trend, but the feature is not statistically significant. These results suggest that resting state LFP coherence is dominated by low-frequency signal coherence, while stimulus LFP response is dominated by high-frequency signal response. But how about the temporal dynamic connectivity? BOLD versus LFP in different frequency bands? Do they share a similar dynamic feature of functional connectivity? That leads to an exploration of a hidden Markov model, because resting state functional connectivity is dynamic. It's reasonable to assume that the true brain states are invisible, but resting state derived functional connectivity that are dependent on the true brain states are visible. So here we have these hidden states. It's actually a Markov chain, which means each state can go back to itself and go to other states. The transition matrix represents the probability of state transitions. And here we have observation sequence. that can be correlation coefficients from two brain regions in FMRI or LFP, calculated using a sliding window technique. And the emission matrix represents the probability behavior of hidden brain states. There are some published papers, and they used Bond-Welch algorithm to find the maximum likelihood estimate of a hidden Markov model, given a set of FMRI signals. But our question is, what exactly are these hidden states, and how do we interpret the results? Therefore, in order to make the model more clear, we came up with our Markov model-based approach. This is the workflow. First, we extracted resting state time courses from FMRI and LFP seeds, and then used the sliding window technique with a window size of 60 seconds to calculate correlation coefficient sequences. The reason for choosing 60 seconds as the window size is based on our previous publication, in which we did several non-model simulations and statistics. Afterwards, we converted correlation coefficients to z-scores, and all z-scores from -1 to +1 were normally classified into 50 states, representing the degree of correlations. Next, we used the two-sample KS statistic to compare BOLD and LFP behaviors in each individual monkey, based on measurements of the distance between empirical cumulative distribution functions for jumps of states, which represent probability of changing states. Therefore, lower KS values between BOLD correlations and LFP coherence can suggest increased similarity in dynamic connectivity. This left figure shows KS statistics values for comparing resting state BOLD correlations and its corresponding monkey's LFP coherence between areas 3B and 1 in 7 frequency bands. Resting state BOLD correlations are distributed more similarly to the low-frequency LFP coherence than the high-frequency LFP coherence, comparing the black and red boxes. This result suggests that the dynamic connectivity of BOLD is closer to that of low-frequency LFP than high-frequency LFPs. Next, we wanted to know how exactly the low-frequency LFP coherence is different from high-frequency LFP coherence. And the right figure shows an example of a sliding window coherence analysis between areas 3B and 1, comparing delta and gamma high bands. Results of 7 frequency bands show that coherence of low-frequency LFP signals change faster over time than that of high-frequency LFPs. This is an ongoing research, and we will also quantify how much faster. Let me wrap it up. Our research aims to investigate the temporal and spatial relationships between BOLD and local field potentials in 7 frequency bands at sub-millimeter cortical modular scale. We found that local functional connectivity of BOLD and the broadband LFP agree in both locations and extents. Correlations of low-frequency LFP signals change faster over time than those of high-frequency LFP signals. In addition, the resting state dynamic connectivity of BOLD is closer to that of low-frequency LFPs than high-frequency LFPs. Therefore, resting state BOLD FMRI signals are reflective of more of low-frequency LFP signals than high-frequency signals. I would like to thank everyone who contributed to this work and NIH funding. If you want to discuss more with me, please come by at my poster at number 1481. Thank you for your attention.
Yes, you got a question?
It was really nice work.
Thank you.
So I'm curious about the lower variability that you saw in the high frequencies, like gamma, because you're using a 60-second window.
Yeah.
Could it have to do with the window length?
How do I decide the window length?
No, I mean, could you see less variability because you're looking at averages over a relatively long time compared to the cycle length?
Yeah, so for a sliding window, it's really tricky because if we use small window size, then most of the variability we saw is actually from the sampling, the limited sampling, because I also simulated VAR model, which is a completely stationary model. And if I use 30 seconds, then I can create those dynamics. So for sliding window, I don't suggest people to use less than 60 seconds.
We'll have a lot to talk about at your poster then.
Yeah, thank you.
Yes?
Yeah, I actually just want to know more in detail how you decide the size of the slide window. So you mentioned that there's a previous work that's
the top right in the human brain mapping paper. So I did VR model, filter stationary model, and non-stationary dynamic model, and I did all the null simulations and used the KS statistic to compare the simulations with the real FMI data, and that's how I decide the window size.
Okay, thank you.
Yeah, we can talk during my poster because if I talk about that, it will be like half an hour.
So if there are no more questions, I have a question. This is about the agreement between the bold resting state and the LFP. So there has been work done on this and suggested that in the resting state, higher frequencies, like for example the gamma frequency, is more representative of of the bold activity. So what's your sense of how to reconcile?
I think so now, if you check all the literatures, there are like two different ideas. One is like the low frequency, like delta. I saw some like PNAS paper from a group, Dr. Liu. And there's another idea is that it's the envelope of the gamma frequency. It's not the gamma frequency itself. It's like the envelope. So from our results, we saw it's the low frequency that is more reflective by both signal. But I can also check the envelope of the gamma and see if the envelope of gamma can be represented by both more.
Sure. Okay.
Thank you.
Thank you.